Provost sir will be joining. Please say success is the some small effort repeated day in day and day out. People who succeed have momentum. The more they succeed, the more want to succeed and want more they win and find a way of success. A very pleasant good afternoon to all of you. Our today's esteemed guest speaker is one of such well-known Indian scientists who has worked day and night consistently, putting up small efforts for decades before getting succeed. He is an inspiration to thousands of your nation. His constant research and development in the area of bioscience is an example of success covered by excellence, endless hard work, and pure dedication with high determination. With this note, I, Dr. Dwani Upadhyay, Assistant Professor, Microbiology Department, with a gratifying smile, feeling honored to welcome our today's eminent speaker, renowned Indian scientist, distinguished Professor Dr. Niaz Ahmed Sir. On behalf of Parul University, we are very truly fortunate to have you with us, sir, today. I'm truly delighted to welcome our head of the department, Dr. Girish Babu sir, and other faculty members and dear students for this distinctive live webinar. I assure you all that experiencing this live webinar will enlighten our knowledge with experimental live learning lessons. I request our Head of the Department, Dr. Girish Babu Kesar, to kindly have some few words on this August occasion. Sir, please.
Good afternoon, sir. Good afternoon, everyone. It's really a great opportunity to have people of your caliber coming over our platform for the webinar and trying to share your experiences, guide us all through to the success. Because maximum of the audience who are there enthusiastically who have been participating into this webinar are the young people. Their minds are going to be really ignited by listening to your words, your experiences, and your guidance, and all that. So it is really a proud moment for us to have you for this webinar, and we are really happy and we are really thankful for accepting our invitation. Sir, we are all very eager to listen to your talk and listen to your experiences also. Thank you so much for that. I request you to continue this webinar, start this webinar. I welcome you for this webinar on behalf of Parul University family. Thank you so much.
Thank you so much, sir. Meanwhile, we are having our Provost, sir, Dr. Emin Patel, sir. Sir, I kindly request you to have few words on this August occasion. Please, sir.
Hello.
Yes, sir.
So, good afternoon. So, good afternoon to one and all. It's really pleasure me to be the part of such talk as our Professor Niaz Ahmed is going to speak on that human-animal ecosystem. It's really a topic of interest because I am the man of technology, but whenever there is a biology and everything comes, I also feel to understand, to learn something. So welcome to Mr. Professor Niaz, and special thanks to Dhoni for arranging this type of talk. Almost I can see that so many talks are arranged by her. It is really a good addition to our all webinars. So my best wishes to all of you, and I hope so that a lot of collaboration will take place and our students will get a lot of new learning, motivation, and rather some of the new ideas, case study might be taught by you, so will be useful to our student for their career. Again, my best wishes to all of you. Thank you. Over to you, Dhoni.
Thank you so much, sir. You are always raising our spirit with your enlightened words. Thank you so much, sir. It is strange that only extraordinary men make the discoveries, which later appear too easy and simple. Our eminent speaker is one of such personality who did extraordinary discoveries which appeared impossible initially in the bioscience. Although Honorable Professor Ahmed Sir does not need any introduction as he himself is an example of success and a proud of India. I would like to take this opportunity to introduce our today's eminent speaker, Professor Niaz Ahmed, is a qualified veterinarian, one health epidemiologist, and professor of biotechnology and bioinformatics at the University of Hyderabad. Professor Sir has graduated in veterinary medicine and obtained further degrees in animal biotechnology. He did PhD in molecular medicine and later he joined the University of Hyderabad as a member of the faculty and served as a chairman of Department of Biotechnology and Bioinformatics. He also served as a senior director at International Center for Diarrheal Disease Research in Dhaka and also as a member of senior leadership team of the center. Professor Ahmed was also affiliated with the University of Malaya, Kuala Lumpur, Malaysia as a visiting professor of a molecular bioscience at the Institute of Biological Sciences. He also served as an adjunct professor of Academy of Scientific and Innovative Research, India. Professor Ahmed Sir served as a section editor of PLOS ONE. Currently, he is providing editorial oversight to PLOS ONE Prokaryotic Genome Collection. He also served as a member of PLOS International Advisory Group and was founding editor-chief of Gut Pathogen published by Biomed Central Limited, London. Professor Ahmed is a member of International Society for Genomic and Evolutionary Microbiology and currently serves as its General Secretary. He is also a member of Faculty of Thousands Biology. He served as a panelist for research proposals submitted to European Commission 7th Framework Programme. He was admitted as a Fellow of the Royal Society of Chemistry and a Fellow of American Academy of Microbiology. He is an elected Fellow of American Academy of Microbiology, the Royal Society of Chemistry UK, the Royal Society of Biology UK, and the National Academy of Sciences India and Indian National Academy of Science. Professor Ahmed is serving and has served as a member of editorial boards of the very well identified scientific journals. He has been awarded with many prestigious awards, including Shanti Swarup Bhatnagar Prize, which is India's highest science award with the hand of Prime Minister in India in the year 2016. He is also a recipient of prestigious National Bioscience Award for Career Development by the Government of India. In 2017, Dr. Ahmed was awarded with the Microsoft Azure for Research Award in Public Health to work towards the genomic of identification of new genes, new proteins, and new pathways underlying the multiple drug resistance phenotypes in bacteria. Professor Ahmed Sir has earlier received the University of Ahmed Chancellor's Award from the Chancellor of University, Dr. C. Rangarajan, the former Governor of the state of Andhra Pradesh and the former Governor of Reserve Bank of India. Dr. Ahmed also holds appointment as adjunct professor at the Academy of Scientific and Innovative Research. He is corresponding fellow of European Halobacter and Microbiota Study Group. I highly feel privileged to invite Professor Dr. Niaz Ahmed Sir to deliver his talk and enlighten us with his knowledge. So we would be honored to know about evolution of pathogen at human-animal ecosystem interface, your research work and experiences. Please, sir.
Should I start the talk now, Dr. Dhwani?
Yes, sir. Yes, sir.
It has been indeed a very flattering introduction. I'm so happy, but it has taken a lot of time. So it in itself has become a lecture. Thank you so much. Really a very big introduction for a small person like me. But nevertheless, I would love to interact with your students and the fellow colleagues and also my friends at the LinkedIn and Facebook who wanted to join me also. They are also welcome. So it's always a pleasure to interact with the online community and the students, particularly with the university students. I am a university teacher, as my first profession, and I love to teach and give lectures. At the outset, I would like to start with the connotation that up to 60% of all human pathogens have animal origins. It's such a superfluous statement, but at some point of its crux, it is true because modified host lifestyle factors have allowed expansion of some of the pathogens that human beings have not seen before. Although these pathogens were dwelling in the animal kingdoms and anywhere or somewhere in the jungles, but then because of deforestation and industrial expansion, those organisms which were once upon a time not known to us, they have made the tropism into among into our societies. Am I audible and visible, as well as are the slides okay?
Yes, sir. Yes, sir. Absolutely.
Okay. Okay, so because of this reality that the hosts of some of the pathogens, like 60% of the pathogens have common hosts, like the warm-blooded animals and the human beings. Also, the terrestrial as well as marine animals. I can give you the example of tuberculosis. Like we were the first group to identify tubercle bacilli in the seals at the genomic level sometime in 2003, when I was starting my career in the area of bacterial genomics. And the seals are living in the both in Atlantic seas, sea and the other oceans, and the seal bacilli, those who are causing the tuberculosis in seals, were so similar to the tubercle bacilli in humans. And some of the seal trainers were also infected, and so the seal trainers isolates and the seals isolates and the bovine isolates and the human isolates were so same. Although they may have never seen each other so frequently because they are in the sea and we are on the surface of the earth and in the cities, and so, but these pathogens are so common as to their evolutionary origins. So all pathogens, they have a predilection, equal predilection, I will see, to the humans and animals. Sometimes it is possible that the humans are accidental hosts for them. The destination is not a human being, but sometimes it is possible that humans are accidentally exposed and get infected, and then the pathogens start liking humans as their hosts. And that's how the disease spreads in the human communities. We know the catastrophic pandemic of the SARS-CoV-2, which has never been a human pathogen, and it is espoused and it is hypothesized, also it has been proved at some stage that this pathogen belongs to bats, and somehow it came in contact with the human beings, and that is how it started liking human as its new destination host, and it has spread so fast with definitely a survival advantage at its evolution. So they spread fast when they have some sort of a advantage to grow. Pathogens, they are looking for some sort of survival advantage. That's very important. And for survival advantage, they have different mechanisms. Why the pathogens would like to survive? It's a basic basic evolutionary propensity or a requirement for any organism to expand its population and to grow and to pass on its characters to its progeny and prolong the opportunities for its progeny to survive, thrive, and live. So organisms, like I will be focusing mostly on the bacterial pathogens in my talk, because that has been the bacteria have been the crux of our research rather than the viruses or fungi, for example. But they are also the pathogens, the viruses, the fungi, the protozoa, and bacteria. So since the bacteria are little bit complex at genomic level, and they fascinate us because it is easy for some of the bacteria to be cultured and handled in the laboratories. So they serve also a good model for the evolution of virulence, for the evolution of metabolism, for the evolution of predilection for certain substrate types. And that's how we prefer bacteria as a model for study. So much of our work is on bacterial pathogens, and that's how I will be talking mostly about the bacteria and not much about the viruses. So the survival mechanisms basically are dictated by their propensity to evolve their genomes and retrofit and get into different pathways so that they could catabolize the available substrates for their carbon requirement. So for the requirement of their energy. So different substrates dwell in different hosts, for example. And so there are definite mechanisms for their survival that they are resorting to. So until now, it is clear that the bacteria are evolving continuously and fine-tuning themselves as regard to the evolving host ecosystem. Every host has an ecosystem, and every host will mount some level of selection pressure on the pathogen. For example, the activated immune responses, the other microbiota that is available in our body, different type of bioactive compounds, bacteriocins, antibiotics, and so many things. They provide a sort of a background for the pathogens to select themselves, as fine-tuned to the changing host environment. So the evolution of bacteria at the genomic level predominantly follows two paradigms. That's what we have been studying and also lecturing over the years and teaching to our students. One paradigm is about the vertical genome reduction. What does that mean? That the genome of the predecessor organism would have been much larger and complex than the genome of the subsequent generations, or the newest descendant will have a smaller genome. And so the order of reduction of the genome has made them more parasitic in their lifestyle as they try to shed their extra genomic burden. That means they have also departed from independent free living pathways and resorted to parasitize upon the pathways of their hosts. And that is how they have become parasitic bacteria, such as the ancestor of Mycobacterium tuberculosis, which is a very dreaded pathogens, which represents a class of pathogens that are very, very nasty. So it causes tuberculosis, which is a very, very complicated disease. After 1964, when the rifampicin was developed, after that, not so successful drug has come in the market. So a huge gap with respect to the effective treatment. There is no effective vaccine, and it kills a large number of people every year. So the latest or the newest species in the Mycobacterium clan is Mycobacterium leprae, the leprosy bacillus. It has a very small genome, something like around 2 MB or less, and most of the genes are pseudogenized. That means those genes are not functional. What does that mean is that this organism is very highly parasitic. That means it cannot independently survive. So it is very difficult to culture Mycobacterium leprae in the laboratory. You can culture it on the mouse foot pad, and that too only few divisions could take place before you can actually have sufficient DNA to sequence or to amplify or whatever. So what I was telling is that one paradigm is vertical genome minimalism, where they tend to shed their genomes over a period of time. And chunks of genomes are lost in such a way that those chunks cannot be regained back again through any mechanisms of recombination. If the evolution is through mutation, the mutations could revert back. But if the evolution is through large deletions, it is very difficult for the organism to acquire that lost genome back, because such large genome cannot be acquired through recombination. So that's one paradigm which the mycobacteria follow, vertical genome reduction. And another, so you can see here, if you can follow my pointer, You see here how the mycobacterial species have lost the successive regions of deletions, like we call them as regions of deletions. And each time that they have resorted to lose a region of deletion in their genome, they have actually evolved a propensity to target a particular host species. So the host species predilection or tropism is the function of their evolving through vertical genome minimalism. That's what I wanted to tell. And another paradigm is through the lateral genome acquisition, through horizontal gene transfer. So the enteric bacteria evolve by this mechanism. They acquire the genes and the gene cassettes from the vicinity or from the organisms that are compatible to recombine when they are in a mixed culture setting, when they are populating a common niche or an ecosystem, and any adjacent or adjoining availability or any donor organisms or species that are available to donate the the DNA material, the genetic material, be it a plasmid or be it a pathogenicity island or an integron or phages for that matter, which are also the conduits for gene transfer through transduction mechanisms. So these recombinational mechanisms, which allow the organisms to aggregate their genomes through lateral gene transfer or horizontal gene transfer, also result into optimization of fitness. And that optimization of fitness is in the form of additional pathways of survival, the additional survival capacity, additional capacity to catabolize new carbon sources, different sugars, different forms of sugars. Okay. But when we analyze them on a population wide scale, these paradigms that they follow for their genome evolution could very well be reflected in their fingerprints. You see the two-dimensional gel images here representative of a population of Helicobacter and a population of Mycobacterium, one representative of the lateral genome, horizontal gene transfer, another representative of vertical genome minimalism. And you see in Mycobacteria, almost the fingerprints are like clonal, because they are losing and losing their genome up to the point that they cannot afford to lose anymore. And so their genomes become almost constant. When their genomes become clonal, that means that gene pool is closed. When the gene pool is closed, that means if you sequence new genomes, you will not find new genes. Okay, how economical. So like if you have a representative genome, that is enough for you to understand the metabolism, the biology, the evolution, the virulence of the organism up to a great extent. But when the top panel, you see when the horizontal gene transfer that is continuously happening, that means the gene pool of the organism, and I exemplify it by the enterobacterial organisms, Salmonella, Shigella, E. coli, Vibrio, Helicobacter, Campylobacter. These classes of organisms, they evolve through lateral acquisition of the genetic material, and that is how their fingerprints are so diverse. That implies that they have an open gene pool, or they have an open pangenome. And when we say that they have an open pangenome, means sequencing of every next organism or every next representative or isolate will give you new genes. So by probability, if an enteric bacterium has an open pangenome, it is possible that every new genome sequence will likely give you 40 new genes, approximately. So new genes means new functions. New functions means possibly new pathways. New pathways means possibly new mechanisms of adaptation. Understood? Adaptation dynamics are important to understand what is their preference for a particular host. If they start liking that host, they may not leave that host. They may create a very tight relationship. and that is how they prevail. So talking about mycobacteria, and we'll end that chapter here and then go forward with the enteric bacteria. So mycobacteria, although they have become more colonal, like you see the lower panel, that most of their genome has now become fixed. Not much variation, but nevertheless some variation is possible that could give you limited diversity or limited flexibility to type them or to classify them into different geographic types. But then this typing is not functionally relevant. It can just give you an idea that if you do this kind of a typing, it is possible for them to geographically classify. And when we used to do this 20 years ago, this kind of a typing, it used to only give us the information as to how closely they are related or how different they are in terms of certain fingerprints. These fingerprints were with the advent of neutral markers such as the intergenic spacers, and they may not have a direct association of any functional pathways or any functional characteristics, but nevertheless it was possible to take the history of the isolate and see whether it was, for example, drug resistant or otherwise, or whether it was possible that a particular resistance will actually associate with a particular lineage of mycobacterium. So it was only to identify their lineages based on neutral markers such as the intergenic regions, and we used to call this technique as a spacer oligotyping, that will only utilize the limited amount of available diversity that is left in the genomes of mycobacteria. But on the contrary, in E. coli, for example, or in Salmonella, or in Shigella, or in Vibrio, or in Campylobacter, or in Helicobacter, these are the representatives of the Enterobacteriaceae. And so therefore, as I told you, that they have an open pan genome. So the meaning of an open pan genome is that if you sequence new genomes, you will find new genes. And new genes means new opportunities to know new pathways, new functions, new fitness advantages, new capabilities to catabolize novel substrates, and thereby new predilection dynamics in order to adjust for the change in the host environment. So the graph, if you see here, is something like, how I can say, something like this. This is the core genome. And this is the flexible genome. So the core genome actually denotes the minimum complement that is required to discharge the metabolic functions. So on the metabolic function side or the core function side, you may not find the new genes. But there is a class which is called flexible genome, where you will find all genes that are novel and required for fashionable things for the organisms to do. And that is how while the core genome is consolidating with the advent of every new genome that is sequenced, or with the addition of every new genome to the gene pool, the flexible genome is always rising. It indicates that there is an open gene pool, or there is an open pan genome for this class of organisms. So that's where I say that every new genome in this situation is actually giving you hypothetically 30 to 40 new genes. It depends, of course, on the kind of organism, the genome characteristics, the sequencing platform, the proficiency with which the genome has been analyzed. Okay. One quick feedback in between. Are you hearing me and am I visible?
Yes, sir. Absolutely.
Great. So now let us move forward. So with these new genes, where these new genes we encounter and what they are doing. So this is the work of our PhD student Narendra Kumar, who is now a postdoctoral scientist in University of Cambridge. He did this work in our lab and published in the journal Nucleic Acids Research some times ago in 2015. What we found is that most of the novel genes in Helicobacter, which is also a enteric bacterium, okay, the member of the Enterobacteriaceae, okay, they encoded a novel class or class of methyl transferases, which were like microbial methyl transferases, and they have a role in epigenetic regulation. So we espouse the hypothesis based on this, that it is possible that some of the microbial genes or the gene products could have the flexibility or capability to regulate their host genome. It's possible that they might regulate the genome of their host, or they might regulate the genes and gene functions of their host in their favor. So it is possible that some of the organisms might upregulate or downregulate certain host genes in their favor. And that is how we were also studying for some time as to how many genes of Helicobacter could actually interact with the human genes. Or is there a gene-gene, protein-protein interaction that might be directed towards a survival advantage to the pathogen? So how many genes are upregulated and downregulated, for example? And whether they have the coordinates, the upregulation and downregulation coordinates have anything to do with the microbial repertoire of such methyl transferases. So I was just giving you an example that if there are new genes that are acquired by these organisms or bacteria, then what could be the possible functions of those new genes? Okay. What additional advantage that those genes could give? In a hypothetical scenario or in a modeling scenario like this, it is possible that these genes may not only give them the survival advantage, but also the capacity for them to also manipulate their host genetic repertoire or the expression dynamics in their favor, of course. And they have a lot of capacity in terms of diversity. It's not like, for example, there's only one gene. Just the femoral gene cluster of E. coli, for example, you see how much diversity is there. This is not our paper, this is somebody's paper. I'm just referring to it. That just the femoral, which are the appendages on the exterior of the cell wall, with which they interact with their environment, with other organisms, the fimbriae. They are also the virulence factors of the E. coli. And in just one class of a particular appendage, how much diversity that is possible for them to create. So there is an unfathomable diversity that is possible. Okay, when the organisms, they evolve, they interact with their environment, they retrofit their genomes. So I am getting the background of a microphone here. Okay. Are you able to confirm that the slides are transitioning?
Yes, sir. Yes. Sir.
Okay.
Yes.
Okay. Then in another scenario, going back to the evolutionary history or the natural history of these organisms, it is assumed that sometimes they have evolved from a non-pathogenic ancestor. And then as they came together with the human societies, they have evolved themselves to become pathogens. We have some time back published this scenario related to the arrival of Helicobacter into the human societies with the arrival of agriculture. And that was because of our motivation to find certain similarities or genetic signatures in Helicobacter which were similar to some of the plant pathogens. For example, the CAC pathogenicity island in Helicobacter, which is a virulence encoding island, that has quite a bit of similarity with the islands that are present in Agrobacterium tumefaciens, for example. These kind of studies require a lot of fossil evidence and other evidences to prove. But then now, nevertheless, this is a hypothesis that as they came in a geographical specific manner, as the agriculture arrived, some of the pathogens have also likewise arrived and evolved, and they inhabited and they became adapted to certain populations in a geographically compartmentalized manner. And today's Helicobacter we see is actually very much geographically compartmentalized. There is a huge body of literature on that the Indian type and Helicobacter may not be fit in Japanese type of host population and vice versa, because they have fine-tuned themselves with the human host population. But nevertheless, some of them, like Helicobacter, Leptospira, they also are very highly capable of infecting a large number of hosts. For example, a Leptospira pathogen, it's very common in Gujarat also. Every year in the monsoon season, we get the cases of leptospirosis. And in Chennai and in Mumbai, you see the lots of flooding is happening, lots of water logging happens. Should I tell you a very good vaccine for Leptospira? Wear long shoes. Very simple. But then there are conditions where people cannot wear shoes while working, like for example in paddy fields. The farmers are working barefoot. The animals don't wear shoes. So the prevention is so simple. Wear long shoes, but there are practical difficulties. When the kids are going to the school, nowadays they are not going. When they are going to the school, sometimes they walk into accumulated water. There is a water logging. So these organisms are capable of penetrating the intact skin through a screw-like burrowing action. These organisms were like Leptospira, they are like 300 plus different serovars, more than a dozen species. These serovars, they evolve dynamically as against the host immune responses. They have a very wide very wide range, from cockroaches to elephants, anything that they can infect. They were endemic to Andaman Islands, but now they have become the mainstream pathogens of the mainland. Why? Because they have been coming in the ships. There are vessels and containers which are moving the food grains, vegetables, and all supplies to the island and the mainland, and these rodents, which are also the host, rats, they carry them and they travel between the mainland and island. And that's how you see the disease once, which was endemic to a remote island location, is now very much encountered in the mainland every year with the arrival of the monsoon season. And if you see the genome evolution here, you see the admixture of the genomic regions. Because not only that they have been arriving here and going back and forth, they are also recombining actively and co-evolving with the local leptospiral population. And that is how the fitness is evolving. I hope it is clear. Coming to another class of, another pathogen, I'm talking about Enterobacteriaceae most of the time, the Vibrio cholerae. The cholera is a huge pandemic that is running. Millions of people have died over the millennia. If not millions, hundreds of thousands of people. The cholera epidemic of the medieval England is very famous in history. And this is a waterborne disease. But when we started to look into the evolution of cholera bacteria, it was imperative to also understand its interaction in the environment. As the topic goes, evolution of pathogenic organisms at the human-animal environmental interface. And you see the cholera bacteria as to how many organisms now it is interacting. Can you see? Marine birds, they come and get hold of the birds, they catch the small fishes. The fishes might have already taken some marine protozoa. Protozoa might have already eaten some of the Vibrio cholerae, and that is how the Vibrio will be taken from these organisms and actually dropped on the soil or the terrestrial locations by these birds. So migratory birds, they carry it wherever they go. So the interaction with the zooplanktons and phytoplanktons and all sorts of mucilaginous substances like fishes and fish scales, their guts, some of the surfaces of the aquatic plants, the blue-green algae, the anabina, and all other kind of vegetative stuff that grows on the surface of the waters, they are the likely dwellings of Vibrio cholerae. When I was working in Dhaka from 2016 up to 2020, the biggest problem there is the seasonal arrival of cholera every year. So the cholera is a seasonal epidemic. Two peaks religiously come, one in early summer, another in late summer and start of the winter. And they come. So when these peaks come, the hospitalizations start. Up to 600 hospitalizations per day happen when the cholera is peaking. And every year it will come. So that means it is endemic in the environment. And you see the life cycle here, that it is having a multi-organismal interaction. And that is how the survival advantage to this organism is enormous. And that is where when they survive, they get into the food chain, they get into the water. When the drinking water is contaminated, you get the outbreaks. In summer months in particular, when there is high humidity and people are working, when they are thirsty, they approach the water sources without much scrutiny of the quality of the source. When the person is thirsty, too much thirsty, he or she may not scrutinize too much the source of water. Not everybody can afford packaged drinking water, and that is how it's a waterborne disease. But you see the colonization factors, the antibacterial activity, all host factors, all microbial virulence factors. Okay, so it will have those virulence factors already in action in its main niche where it is dwelling, the marine habitats, the water habitats, and also in its accidental host, that is the human being. The same kind of machinery is operational, but how much flexibility that it has, how much fitness advantage that it exercises. So much of arsenal in its factory. You see, different type of toxins, the different type of adhesion factors, the different type of chemotactic factors, type 6 secretion system, the Colix toxin, the cholera toxin, okay, the VSP locus encoded genes, the chemotaxis factors, so on and so forth. I mean, I do not want you to go into these intricacies of the virulence mechanisms, but just see the flexibility of the organisms, these organisms to discharge or to secrete or synthesize these virulence factors at a particular stage as fine-tuned to the available host niche in the vicinity so that it can expand its tropism relentlessly. Okay. Another mechanism for it is to break the quorum, to break the control of quorum, to break the control through phages, for example. The bacteriophages are the viruses that they attack the Vibrio cholerae in the environment. But when these organisms are in large quantity, when large numbers, they communicate with each other through the quorum sensing mechanisms. That is also a fitness mechanism. We are talking about the evolutionary dynamics as regard to the fitness advantage. So the quorum sensing capabilities are also there apart from the virulence factors that we are seeing here. Apart from these virulence factors, there are other social factors such as the quorum sensing. Quorum sensing is the function of certain molecules, which are the signaling molecules. But then when they sense that there are enough of a quorum, okay, then they will defend themselves against the infecting also. So the phages which will infect and kill them, they will be overwhelmed because of this behavior. And when using quorum sensing, some clever bacteria, they survive and they grow to cause the subsequent seasonal cholera epidemics. And you'll see that when the quorum sensing mechanism is at its peak, when the phages are not anymore to control the emergence of Vibrio cholerae, then suddenly there will be an outbreak. And you'll see that the hospitalizations are happening. Nevertheless, when these organisms are so flexible and complex in terms of their interaction, there is a lot of role for the flux, for example, the environmental flux, the waterlogging, when it is raining, when it is waterlogging, when the phytoplanktons, they bloom on the water surfaces, how much is the rainfall, how much is the humidity, how much is the population density, how much is the rate of population migration. So the virulence factors, the environmental flux, the soil and water parameters, the population dynamics, all these things could be put into a model. We have a lot of acumen for computational superiority. We are a computational power. We have so big, computational industry in this country and everywhere else. It is possible with the advent of computational programming, artificial intelligence, and machine learning to have diverse data integration as regard to these epidemics and infections, so that the infections or the outbreaks could be predicted, could be computationally predicted, that whether there will be an outbreak this year, if there will be an outbreak, how much will be its severity, in which month the outbreak will start, by which time the outbreak will end. And so it will be a kind of a help for the health planning authorities, for the healthcare machinery to gear themselves, to take their resources together in the anticipation of this kind of. I'm not talking about Dhaka city or any other city like Kolkata, where the cholera is endemic, but I am talking about where it is possible. Phytoplankton blooms on the sea in itself is a marker or an indicator. These blooms can be captured. These blooms could be captured on the satellite. That if there is a larger bloom this year, there is a huge bloom, heavy bloom, there could be a possibility that this year there will be an outbreak. So these kind of things are under testing now. We have a overwhelming interest in these kind of algorithms, but we are also using it for some other pathogens in context of some other phenotypes. Like for Vibrio cholerae is very easy to manage. It is very easy to control, provided you get rehydration done in the given window. Although 600 people will be hospitalized, but there will not be much mortality, because people are aware by now in that country, at least, that they should give oral rehydration to the patient as soon as possible. And by the time he or she is brought to the hospital, then there will be saline given, and in 2.5 hours, the patient can be discharged. But there could be very serious problems in other organisms which are also of the enteric class, that they have phenotypes such as the multiple drug resistance. And when multiple drug resistance is there in some of the organisms, it is very difficult to treat. Therefore, in such organisms, it is imperative and it is necessary to also understand what are the host and pathogen factors that are responsible, and how to study these organisms as regard to their epidemiology. Epidemiology is very, very important, because with epidemiology, you tend to understand the infection and spread, the transmission dynamics of the infection, as to whether the infection is limiting itself or it is augmenting. It is increasing or it is decreasing. It is under control or it's going out of control. So good olden days epidemiology was done with the help of neutral markers. As I told you, that if you can go back, I can show you the Mycobacterium tuberculosis epidemiology that we have done in 2005. You see here, it was done with a limited number of markers that were the neutral markers, and there was no functional handle. That was the epidemiology of good olden days, done with the help of neutral markers, like spoligotyping or the intergenic regions. So here you see the classical molecular epidemiology, tagging and tracking of infectious pathogens with neutral markers such as insertion sequences and intergenic elements. It doesn't have a functional, handle. It cannot tell you whether the organism is adapting or whether it is, having any disease phenotype that is different than what has been mentioned in the textbooks. Okay. But then functional molecular infection epidemiology on my left hand side, on my right hand side here, you see that there is an emphasis on phenotypic and genotypic correlates, because now we are in the era of genomics. And this epidemiology is done with functionally relevant markers, the markers that have functional relevance, such as the genes that encode antibiotic resistance, the single nucleotide polymorphisms or mutations that give the bacteria a gain of function. Multiple drug resistance could be because of the gain of function, loss of function also. Some mutation could give them a selection pressure or a selection for a particular phenotypic trait. Okay. And this is relevant also when we juxtapose these evolutionary signatures with the host. The host is also evolving. The pathogen is also evolving. The host also not only that host has its own genes, but host also has its resident microbiota. We call it as, microbial consortia that are inside our body. Around 14 trillion different bacterial cells. They inhibit our body and make our personalities more like microbial personalities. We are less human than microbial, so we are more microbial, but not all those microbes are pathogens. They are our resident microbiota. And so our genes and resident microbiota are in a dynamic interaction with the pathogens at a given time. And so what are the coordinates of that intricate interaction over a period of time, over an evolutionary time scale? And that is called as functional molecular infection epidemiology. Okay. Epidemiology is more complex, more functional, more relevant, more effective, more meaningful, more productive nowadays. Okay. That is how we have a working definition of epidemiology now, that functional molecular infection epidemiology not only implies descriptive host pathogen associations for or risk analysis, but rather the interplay between the pathogen and the host genomic variations to functionally demonstrate the role of the genetic variants during infection. And you see as to why epidemiology is the science of so high importance. And now in the COVID times, you see now everybody is realizing the magic of epidemiology. So now the government agencies, everybody is keeping a watch on R0. What is that? The basic reproduction rate at which the virus is multiplying and virus is infecting other hosts. That's very important. Okay. And when we imply epidemiology in the context of survival mechanisms of bacteria, the greatest survival advantage that they could have, the fitness advantage is the multiple drug resistance. And the multiple drug resistance is like when they have resistance to more than one antibiotic. So the treatment options are limited. But the bacterial flexibility to adapt, to evolve their genomes, to retrofit their genes, and to diversify their functional acumen is ever increasing and flexible. And that's how we have this emergence of MDR pathogens. So the evolution of multiple drug resistant bacteria is actually bacterial genetic driven and environmentally driven. And it is also a function of the industry. So it's a multi-sectoral issue. Why I say like this is in this slide you see. the antibiotic overuse in animal farming, because antibiotics are traditionally given as supplements. When I was a student of veterinary medicine, something like 20 years ago, or even before, these things like were very much part of the animal ration. The antibiotics, chloramphenicol. And so the birds and the animals used to be fed with this for the weight gain. I don't know what has happened over the years to the resident microbiota of those farm animals. That's a different story. We'll talk some other day. But you see, the emergence of multiple drug resistance as a fitness advantage is actually a function of indiscriminate use in both the sectors, livestock sector as well as the human medicine sector, either due to non-compliance, also on the part of patients, because they sometimes do not take the full course of antibiotics. People tend to self-medicate themselves. There's a discharge of antibiotic residue from the production units into the environment, or there is a possibility that the animals that are carrying the residues in the meat from the farms. But nonetheless, with genome sequencing, it is possible to document what are the coordinates of such fitness advantages. Whole genome sequencing could give you the handle on understanding the behavior as to genome evolution over a period of time. And over a period of time, if you see that AMR in common pathogens is rising exponentially. Now, Klebsiella pneumonia, for example, an enteric pathogen, is 100% resistant in most of the settings to all available antibiotics. Can you imagine? If there is a hip dislocation, there is a major surgery, and if the surgical wounds are contaminated with Klebsiella, it spells doom. E. coli is such a common pathogen, causing mastitis and production-related problems in dairy production, dairy factories, dairy herds. Mastitis is a big problem in animal industry, and E. coli is the main culprit to blame. Infectious infertility in animals, wound infections in humans, the kidney infections, the uropathogenic E. coli, the enterotoxinic E. coli, the meningitis causing E. coli. So E. coli is such a common pathogen, and it is getting resistant to 100% of the antibiotics. So these are some of the problems. Antimicrobial resistance is the pandemic which is next to the COVID-19, and it is postulated and predicted that AMR could result in 50 million deaths in the next 10 or 15 years if it is not controlled. So to control antibiotic resistance is not by administering some vaccines. It's to change the policies. It's to rationalize the use of antibiotics. It has something to do with the production of antibiotics. And it has something to do with the identification of new antibiotics. Also, for repurposing of the old antibiotics, for re-engineering of some of the organisms to forget the memory of antibiotic resistance and revert back. These are some of the fascinating possibilities, but I don't know how much practical they will be. But nevertheless, it is possible for us to discuss this, keep discussing as to how big this problem is. And at the base of this problem is the evolution. And for evolution, you need a selection pressure. And that pressure is being provided by us. The organisms are selecting themselves as against the evolving human societies. As our lifestyles are evolving, they are evolving. So now it is up to us as to how we want to stop this. So there are so many things to talk, but the time is limited. Therefore, we'll stop here and we'll ponder as to what we have discussed over this 1 hour period of time. Bacterial pathogens, they evolve and retrofit their genomes in response to the evolutionary constraints, and they gain fitness advantage in different settings. Genomic analysis offers a robust tool to glean new genes, new functions, and new lifestyles that are shaping up in different hosts over a period of time. And the AMR is a gigantic multi-sectoral issue that needs to be urgently addressed. At the end, I would like to acknowledge the institutions at which I have worked, my past and present PhD students, as well as the collaborators and some of the grants. And thank you so much for your attention. I always like to give lectures at the universities, and hopefully it was clear without any interruption.
Thank you very much, sir. We are really fortunate to hear you in person. I'm sure all the viewers are motivated, encouraged, and gained a lot from your descriptive and illustrative session. Few of the viewers, sir, they have asked us silly questions that they are more interested in knowing about your work. So they had requested me that, Madam, please ask sir about the experience when he was working on a molecular infection epidemiology and genome sequence-based evolutionary dynamics of the zoonotic and foodborne infections.
The experience has been overwhelming. That's how the experience basically drives you forward. You learn. I started this kind of work when I was a master's degree student. I was given the topic to work on the detection of mycobacteria in dairy cattle, and how to approach cattle when you want to detect mycobacteria, whether the cattle has tuberculosis or not. They may not cough sometime. Cough we cannot identify, but it is a silent disease. And that's how you have to look for a very pertinent sample where you can actually capture the coordinates of infection. And it could be milk, it could be urine, it could be nasal secretions. I used to work with the dairy cattle, dairy herds. I was a veterinarian. And that's where I actually developed the interest as to how to culture, analyze these pathogenic bacteria. Then came the genome sequencing. Then there, how to sequence, how to interpret, how to analyze, and how to link it back with the human population. The thing is that everything is for the benefit and welfare of the human societies. Animals are also working for them. Scientists are also working for them. Everybody is working for the benefit and welfare of the human society. So if you have that type of motivation, whether you are working in an animal sector or you are working in a human hospital or wherever, the problems are common. So working with bacteria is very fascinating, as I say, is that it's very easy as a model. They are very easy as a model because you can culture them. They can grow very fast. E. coli, for example, you can just inoculate your culture in the morning, and in the afternoon you have the needed turbidity to start your work. It is so fast growing, and that's how it is sometimes very fascinating to work with these models, with these organisms. With no other organism you can get faster results than the bacterial organisms as a model. And you can actually run your sequencing in the evening, and by the time you finish your dinner, your results are ready. Before you go to the bed, you can analyze, have the preliminary idea in your mind before you sleep. So it is so fast, and that's how it is so fascinating.
Thank you so much, sir. There is one more question. Uh, do we have any synthetic method by which we can stop the mutation in bacteria?
Of course, there are already published evidences how you can reverse engineer some of the organisms in the laboratory to make them sensitive back again.
Yes.
It's possible because people are working on different substrates. It will be a very big invention if you develop a substrate or a media on which you can grow the organism such that it can revert back to its sensitive phenotype. But then in the field, how it will be working out, it's very difficult to say. But nevertheless, laboratory level efforts are on to reverse engineer some of the mutations to make them sensitive again. So there is a concept of using different antibiotics together as a mix, or repurposing of the previous antibiotics and so on and so forth. So there is a cross resistance. Also, there is a kind of complementary phenotypes or those kind of things. They are already happening.
Okay, sir. Thank you so much, sir. Which advice would you like to give our young students who are research enthusiastic and pursuing their UG/PG programs in bioscience?
The advice to the student is that the pursue the voice of your mind. I don't know
Yes.
if mind has a voice, but what comes in your mind, what interests you, that you pursue. You should not like be carried away by something and then say later on it may not interest you. So pursue your interest and do not look for shortcuts. Give your full potential, give your complete resources, your dedication to the problem. And hard work is the only shortcut to success.
Thank you. Thank you so much, sir, for sharing your deep knowledge with us. You are an inspiration to all the young students of our nation, and your research is a motivation to young science intellects. As despite so many odds and obstacles, you constantly work with a high spirit and enthusiasm to achieve the goals. I'm so much thankful to you. I would request our head of the department, sir, Dr. Girish Babu, sir, so kindly provide us few words. Thank you, sir.
Sir, thank you so much for continuing that kind of a complexity what I have been having relating to the pathogenicity and the genomes of those organisms, which are these days it has become really a big thing, which always would be revolving around mind, and I would be looking into the sequences of the genomes, their hotspots, are the varying regions, are the specific markers which are going to be correlating to the pathogenicity of those things, and many of the things into it. I really got lots and lots of insights when I was looking into these particular kinds of the informations all through these years, and hope so like the other audiences who have been with the webinar, they also got lots of informations and the insights for their queries what they had. And as I was expecting that a person of your caliber would always suggest one thing, like you need to always go behind your dreams without compromising, and at the same time never try to look for any shortcuts, because there is no shortcut for any greater success as it is. Sir, it has been a great pleasure. We really had fantastic sessions with lots of insights, and we really enriched our knowledge in genomics and the pathogenicity as such. So I take this opportunity to thank you on behalf of a very big family of Parul University. And we wish to look forward to work with you, sir, actually, because we are extending in our research wings also. We are going into lots and lots of the different areas of biological research. So I would be looking, we all would be looking to, work with you, and we need your guidance and your encouragement and assistance into the various features. And at the same time, on behalf of Parul University, I take this opportunity to invite you to visit our university sometime on your comfort. We will be in touch with you, and we would be very delighted to have you in person here in our campus. Thank you so much for joining us, sir. Thank you so much.
I look forward to. You're
Thank
most welcome, Girish and Dhwani, and we look forward to interacting with you face to face.
yes,
Sure, sir.
Hopefully very soon. Hopefully very soon, and there is no end to the collaboration. Collaboration is always welcome. Thank you so
Thank
much,
you, sir.
and very
thank
good
you, sir.
afternoon.
Thank you, sir. Have
Thank
a
you.
good day.
Thank
Thank
you
you so
so
much.
much. I would like to extend my deepest gratitude to Giri sir as well, and to all the listeners and all the viewers. Thank you so much, everyone. Thank you so much, sir.
Thank you, sir.